Okay, I think we're going to go ahead and start, and I can see it's a full house here. Um, I would tell people to come in and sit in more on the inside, but it's totally crowded in there. So I'm sorry about this. This is the only room that was available for this talk, and I thought getting a much bigger room was going to make it a little bit less intimate. So this is the best we could do. All right, so it's my great honor, and it's actually sort of my challenge to introduce Maynard Olson tonight. And the reason it's both an honor and a challenge is because Maynard's had such a prolific career that it's really pretty hard to know where to start in terms of talking about his career highlights. But Maynard's actually made it a little bit easier for me because I asked him to send me a copy of CV, and that was overwhelming. But he also sent me a really short narrative, which made it really great because it hit all the highlights. So I'm basically gonna use that in my introduction. So Maynard's undergraduate career was at Caltech. He got his PhD in chemistry at Stanford. He then joined the faculty here at UW in 1992 after short faculty careers at basically Dartmouth and Washington University. He's currently professor emeritus in the Department of Medicine and Genome Sciences. And the reason you guys are all here is because Maynard's actually very well known for his contributions to the Human Genome Project. And in particular, Maynard served on the original National Research Council Committee on Mapping and Sequencing the Human Genome and the National Advisory Council of the National Human Genome Research Institute. He also testified about the Genome Project before congressional committees. And through these activities, he basically played a major role in helping to shape the strategy and the direction that the Human Genome Project went. And then more recently, he served on the National Research Council Committee on Precision Medicine. And I think he'll probably talk a little bit about his thoughts on that topic tonight. In addition to these activities, Maynard's lab also played a really instrumental role, a set of roles in developing some of the strategies that were used to map and sequence the human genome. So as a result of these contributions, Maynard's received many awards and many honors over the years. I can only mention but a few. He was elected to the National Academy of Sciences. He received the Genetic Society of America Medal in 1992. The City of Medicine Award in 2000, the Gardner International Award in 2002, and the Gruber Genetics Prize in 2007. So lastly, I also want to mention, in addition to his international contributions to the Human Genome Project, Maynard's also had an enormous impact on the scientific community here in Seattle. In particular, I think he was one of the major architects in the formation of the Genome Sciences Department about 10 years ago. And in fact, if I'm not mistaken, I think he's actually the one who coined the name of our department, Genome Sciences. And so basically, for those of you old enough to remember the old E.F. Hutton commercial that always ended, when E.F. Hutton speaks, people listen. To my mind, that's kind of how I think Maynard is kind of the genetics version of E.F. Hutton. I mean, when Maynard speaks, people listen. I think if you know Maynard, you know that I'm right about that. However, now I don't know that much about E.F. Hutton. But Maynard is by no means a one-dimensional guy. If Maynard has thought about a subject, doesn't matter whether it's genetics or ballet or you name it, he's going to have something interesting to say about it. He's a really fascinating individual. It's always interesting to listen to him talk, and we're really lucky to have him here this evening. I don't have to repeat his title; it's right up there. So without further ado, Maynard Olson.
So let's try out the microphone. Thanks, Leo. That's a challenging introduction to follow, and this is certainly a challenging audience. It is intimate circumstances. Make yourselves as comfortable as possible. Sit on the floor, whatever, at least until the fire marshal shows up. This is an extremely challenging audience to deal with, since it has everyone from the chairman of medicine and genome sciences, many of my senior colleagues in it, to high school students and members of the public who took seriously the department's sort of advertisement that these lectures are accessible to people without any prior background in genomics or other areas of biology. So spanning this range will be challenging. How many high school students do we have here? Quite a good group. So that's great. I don't have a lot of opportunities to talk to high school students, but I'll try to direct a few of my comments in your direction. And I'll count on my colleagues to bail me out if I get in trouble. I'm amongst friends, and there are people here who know more about any particular thing that I'm going to talk about than I do. So, financial disclosure is on the Scientific Advisory Board of Illumina, which, as many of you know, is a market leader in providing genome sequences, genome sequencing instrumentation and reagents. So I'd like to dedicate these comments to my colleague, Arno Matulski. He had hoped to be here this evening, but was unable to be. And perhaps for the high school students, I'll just say a word or two about my interactions with him. First of all, young scientists often are concerned about what they should major in, what they should focus on. A lot of choices to be made at a stage when you don't know too much. And I'd like to reassure you there's always time to learn. I have no background in medicine, not much in biology, but there are many opportunities in rich university environments to expand your skills. Arno has been my primary mentor in medicine, starting in the mid-1990s. I had lunch with him a couple of times a month, went on for 15 years, and over the period I gradually learned a few things about medicine from the master. So when he was interviewed a few years ago in the New York Times, he gave one of his typically skeptical comments. What we know about the genome today is not enough for all the miracles many expect from this field. Arno and I are one of our bonds, as we're both skeptics. The situation is, however, changing and changing quite rapidly. In the last, I would say, 2 years, a clear corner has been turned in the interplay between genomics and medicine. There's been a lot of discussion of the interaction between this basic science field and the clinic, but now you're actually starting to see serious engagement between genomics and medicine, and so tonight I'm going to try to provide a basic scientist's view of what that's about and where perhaps we could go from here. As Leo indicated, sort of on a policy sense, my background goes back to the early days of the Human Genome Project. I served on this committee, which was chaired by Bruce Alberts. And I'm going to talk a little later about the sort of hype problem that genomic medicine has suffered from the beginning. But the beginning was certainly not this report, which was an influential sort of basic outline of how this project might proceed and what the rationale was for carrying it out. This, I think, was about the most extravagant statement in the entire report. I think that there's no question that the Human Genome Project, it's been evident for a decade or more that it has greatly enhanced progress in human biology. It's indisputable. And so my message tonight is that it is actually starting to deliver progress in medicine. But I think that there are many challenges, scientific, clinical, and policy-wise, to take advantage of this sort of turning point in the kind of history of basic science and medicine. So, taking seriously the notion that we need to get back to the fundamentals of biology, simply using this drawing, which I did myself. Well, remind people that most human cells contain two nearly identical 3 billion long strings of the DNA monomers, G, T, A, and C. These, I say, are nearly identical because in a sort of typical outbred population, such as ours, they're about 99.9% identical. And much of our discussion tonight will involve the minor differences between different instances of the genome. As far as a very basic biology background goes, some of my comments will require a sort of a basic understanding of what genomes do. This is a standard textbook schematic. And the really key point is that the double helix at the top, the genome, is this long string, GTA, G, C, 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 T, and so forth, 3 billion letters. That it has a rather complex organization, as indicated by these exons, introns, and other features. There are quite complex mechanisms that are involved in implementing this information, largely in the form of protein sequences and protein structures. So the sort of basic background that's required tonight is that the order of these monomers in the DNA polymer specify by a complex set of processes that are reasonably well understood compared to much of the rest of what I'll have to discuss tonight. The order of the more chemically diverse monomers in proteins, and it's proteins that are the sort of main functional elements in cells that make up the sort of structures and biochemical functions in cells. I am going to say a few words toward the end of the talk about drug development and the interplay between genomic medicine and development of new pharmaceuticals. And there, really, the main lesson adequate for tonight's discussion is that most small molecule drugs, which is what I'm going to be talking about, drugs that come in pills, usually taken orally, most small molecule drugs work by interfering with the function of particular proteins in cells. The small molecules bind to the proteins in some way that interferes with their function. An important point whose significance will become clear as I go on, is that most mutations, sort of random changes in the genome sequence, if they do anything functionally, also interfere with the synthesis of proteins at their appropriate levels or in their appropriately functional form. And that parallel is something to keep in mind as we dig deeper into how it is we're going to use genomics to improve health outcomes. So a key point in this sort of view of genomic medicine is that the whole endeavor is about interpreting these idiosyncratic features of an individual's genome sequence in the service of improving medicine's sort of traditional goals, prognostication, diagnostics, and therapeutics. So the problem to a bioinformatician looks something like this. So these are real data in deference to Mary Claire King, who's here tonight. This little chunk of the BRCA1 gene, which she no doubt can recognize. This is a little over 1,000 of the GAs, Ts, and Cs, and the gene as a whole is more like 100,000 of these, but I'm sure she knows them at least in their common order from memory. And so I've colored a C out in the middle there, red, just to set the stage for genomic medicine, is that one is constantly looking at a sequence which is nearly identical to one that you've seen before, but has some odd change. Let's say that most people have a G there, and this person has a C, and the question is, what significance does that have for human health? So, for example, for the high school students here, I encourage you to tackle problems of this type with an approximately AP level of just biological background. You actually could go on the web and come up with a rather intelligent estimate of whether that alteration was likely to increase the susceptibility of the individual with this genome to breast cancer. And if anyone wants to take up this challenge, send me an email and we see if we can organize a virtual study group. You can find my email address easily, and in fact, if you can't find my email address, you need to build up your web searching skills, because it's impossible to be a genomicist without being good at searching the web. Well, it's clear that in most instances, the weak link in this rather abstract structure that I've outlined is our primitive understanding of human biology, particularly at the molecular level. A lot of classical medicine was built up at the level of anatomy and physiology and development, sort of organ, tissue level function, and much can be done. But the therapeutic interventions that will be of main interest to us occur at the molecular level, and the issue is whether we understand enough about the molecular processes to make progress. I think it would be fair to say that one reason that surgery has advanced so spectacularly during my lifetime is that these advances could largely be driven at the level of anatomy and physiology and pathology without much knowledge of what the molecular details were. We now are attempting to move sort of molecular medicine more into the center stage. This is not going to happen rapidly, but I actually believe it will happen, and will outline my thoughts on that subject. So to a significant degree, genomic medicine is actually attempting to bypass our ignorance of precisely how particular changes in the sequence affect health. That's an important point. It may seem trivial, but it's an important point and one to keep firmly in mind. There always are two alternatives in medicine. The one that we use as a sort of a mental construct of how this whole system is supposed to work is that we study all of the molecular details and understand the biochemistry in great detail, know what molecules are interacting with which others, develop rational kind of ways of intervening. Or there is some kind of workaround that we don't have all of that information. The latter approach has accounted for most success in medicine and will count for most success going forward, I believe. This is not to say that all that mechanistic information isn't useful, and indeed, I think one of the very broad spectrum points about genomics is that it allows us to leverage what molecular mechanistic information we have spectacularly. And that, I think, is one of the principal lessons of the genomic era. Now, anyone who learns that some process that he or she is studying is affected by a particular little bit of DNA sequence in the genome, the first thing that one does is to find, well, where does that sequence or sequences like it occur in biology? What RNA molecules, what DNAs, other places in the human genome or in other species occur? And the hope is that somewhere in this web of similarity, you'll come to some solid experimental evidence that this is involved in coding for a protein that does this or that or the other thing, and then work by analogy, or what evolutionists call homology, often more powerful. That is, that you assume this similarity as a reflection of long-conserved evolutionary processes. But nonetheless, keep in mind that this is not a sort of system biologist's dream of having highly predictive models of how cells work and so forth. This is an effort to essentially bypass the extreme difficulty of this problem. And this problem, I firmly believe, will still seem extremely difficult 100 years from now. This is not a problem that lends itself to sweeping resolution. So, I've already indicated that each person's genome contains a lot of variation that we know little or nothing about. Indeed, the VUS is kind of the central player in genomic medicine, a variant of unknown significance. And as clinical reports now start to come back, they're full of VUSs, and that's, of course, not what lab medicine physicians want to put on their lab reports, but we have to learn to live with them. I just used an example of an anonymous Korean individual where the genomics would happen to have been done with particularly meticulous care, and in this instance, which was fairly typical of what you'd see in other populations, other individuals, that there were 3.5 million differences. These mostly are substitutions. There are other things, but they're mostly substitutions. Relative to the arbitrary human reference sequence, which can be taken to be more or less a representation of one arbitrary human genome. And about 500,000 of these had never been seen before. And over 10,000 of them were predicted to actually alter the sequence of the proteins that are made in this individual. And so this just gives an idea of the rather massive amount of biochemical individuality that is out there and certainly affects people's health. These numbers are not changing particularly rapidly. The number that have never been seen before is sort of dropping slowly as we see more and more, but theory and experience both predict that that number is going to stay high for a very long time. So, should really pause for a moment because things are now going to get a little more intricate to see if there are questions at this level. I've tried essentially to outline a point of view toward my material so far, and then I'm going to tackle the material. Yes?
That was 3.4 million out of how many, what length?
6 billion. So these are the two genomes in this individual, the maternally derived and paternally derived ones. And so it's, we're talking about a little less than 1 in 1,000 of nucleotides in any particular genome. Monomers are different. And so my BRCA1 just example was to indicate roughly the density at which variants, mostly of unknown significance, appear in genome sequences. Although once we get some of our students to work on that case, its significance may become clear. So I'm going to tackle this large landscape in 3 parts with a look at diagnostics, and then at therapeutics, and then at policy. So I'm going to start with some comments about diagnostics. Diagnostics is traditionally thought of as the strength of genomics because indeed it's where the idea comes from that genomic knowledge is sort of dangerous. It has an oracle-like character that one can peer at these G's, A's, T's, and C's, not know very much, as I've emphasized about why putting a T in this position has some particular effect, perhaps offset by many years, but experience can show that it does. And so there's been a worry since the Alberts report that genomics was mostly going to serve to widen the gap between diagnostics and therapeutics. Diagnostics, as I'll emphasize, is obviously critical to medicine, but physicians don't want to be in a position of diagnosing all kinds of things that they can't do much about. And this is a burden of genomics, is that it has widened the gap between diagnostics and therapeutics. And I think that policy-wise, this is an issue that requires very close attention. So, from the precision medicine report, about which I'll have more to say later, we acknowledged firmly that diagnosis is the foundation of medicine. I think I wrote this sentence: accurately and precisely defining a patient's condition does not assure effective treatment, but it is unequivocally the place to start. No one can argue much with that. And so the subtitle of the precision medicine report was sort of toward a new taxonomy of disease, a new way of classifying diseases. And we envision this world with new taxonomy, more accurate diagnoses, targeted treatments, improved health outcomes, and so forth. Should think ambitious thoughts, and just because there are many difficulties is not a reason not to be ambitious. So let's start with what I think can fairly be called a poster child story for genomic diagnostics. Some of you may be familiar with this. This is the story of the Beery twins. His family has played an important role in sort of waiving their right to privacy and being public advocates for more work of this kind. I heard the mother of these twins speak at a scientific meeting last fall. So this is a story that mostly unusual in the happy outcome, and I don't want to imply that it's in any way typical. But on the other hand, we need success stories in our society and medicine and so forth, and they have a large impact, and they have a large impact for a reason that one shouldn't be cynical about. It's a positive reason. So the Berry twins had a difficult start in life with missing their developmental landmarks, having muscle contractions that were not controllable, unclear that they would be able to walk, particularly the girl was the more affected, with this very aggressive, persistent mother. At age about 5, it was discovered that they had a form of dystonia, this muscle disease, which was responsive to dopamine, which is a well-established therapeutic in the central nervous system. And they both responded rather impressively to dopamine and were able to proceed in school and have a relatively normal early childhood. But by the time they were roughly the age shown here, both of them were encountering kind of a new layers of difficulties. The girl in particular often had difficulty breathing and was unable to exercise vigorously and so forth. The boy was losing fine motor control. And so the mother went on a sort of a new quest that ended with genome sequencing at the Baylor Genome Center. And they discovered that the problem with these twins was a mutation in the so-called SPR gene, which is upstream from dopamine, and it explained why they had been responsive to dopamine, but also upstream from serotonin. And so a new therapeutic could be fed literally into these kids, which was just below the block in this gene, and they both responded spectacularly to this treatment, as indicated in this picture, and are leading the lives of normal teenagers. So there are some lessons here. One is that never underestimate the importance of diagnostics in pediatrics, including in discouraging and difficult pediatric cases. There have been many studies. Talk to any pediatric geneticist. Had lunch today with Anita Beck, my former PhD student, who's a board-certified pediatrician and geneticist here at Children's, and she reconfirmed that you talk to patients of children that have congenital difficulties, and the parents want a diagnosis. They're not under the illusion that any high fraction of these are going to be another Berry twin story, but they want a diagnosis. So some of the basics here are that there are about 4 million live births in the US alone, and this is by no means a uniquely American problem, but starting at home. Medically significant congenital problems occur in a few percent of these newborns, depending on how tight the criteria are for judging a problem to be medically significant. So we're talking something on the order of 100,000 children a year that come to fairly serious medical attention for what appears to be a problem present at birth. And diagnoses are often elusive, and particularly at a level of detail adequate to guide treatment, establish prognosis, estimate recurrence risk for the parents, and so forth. That is, many of the diagnoses, autism, just for example, are in one sense well-established medical diagnoses, but they contribute essentially nothing to guiding treatment and establishing prognosis and so forth. So this is one large target audience that I think we will see much more intensive use of genomics to increase the diagnostic success rate. So this leads, of course, to the question of how often could genome sequencing help? Well, in a current review of this subject, an annual review of genomics and human genetics, there's this kind of brave statement. I like brave statements. Informal estimates estimate that it would assist diagnosis in something like 1/4 of cases, improve management in perhaps 3%. and lead to greatly beneficial treatment, maybe 1%. So I was interested in where these numbers came from, and they were referenced to a New York Times article in which Gina Colada had gone around interviewing people, undoubtedly including the authors of this review, as to what they thought these numbers were. Obviously, there are serious problems with both the numerator and the denominator in coming up with these numbers. Nonetheless, I've been to enough meetings now, like the Personal Genomes meeting that Evan Eichler was one of the organizers of last fall at Cold Spring Harbor, and a Gordon conference I was just at in Rhode Island, where a lot of unpublished data was presented by people from academic medical centers that are sort of starting to do this. And I think these numbers actually are something like in the right ballpark. That is, there is diagnostic success in a significant fraction of cases, a minority, but a significant minority. And there are an accumulating list of examples in which it has sort of direct clinical benefit. Just one quote from really a primary research source. Howard Jacobs, a rat geneticist, a very basic scientist who's become passionate about this subject at the Medical College of Wisconsin, published again this year, Science Translational Medicine, sort of an overview of how it was going there as they've intensified their work in this area. And so they are getting a significant number of diagnoses and, that are considered solid. And an important point is that, and this theme comes up repeatedly when you talk to people engaged in this type of work, is that they initially have concerns about cost, data accuracy, the quality of the sort of annotation that can be done, all these variants of unknown significance and so forth. But the major challenges, in their opinion, and I've heard this repeatedly, relate to how clinicians use the data and how patients and their families deal with secondary and incidental findings. This is a major issue in this subject. It's actually a rather major issue throughout medicine, but that's a topic for another night. We do all these lab tests, we do all this radiology, and we find out all kinds of things that are unrelated to the presenting complaint of the patient. And it's not easy to know what to do with this information. We often don't even really know what it means because the very same radiological lesion that shows up in someone who came for treatment because he had been spitting up blood and had shortness of breath has a different significance than in a patient where you happen to catch a glimpse of it when you were checking to see if they had appendicitis. And that requires immensely more data and data of a different kind and so forth than we have. Well, the genome has been correctly pointed out to be sort of the mother of all sources of incidental findings, because you may be attempting to understand some particular, say, muscle disability in a newborn, but you find all kinds of other things. And what do you do about that? So there are challenges. And at the end of the talk, when I get back to policy concerns, I'll say a word or two about them. So pediatric diagnostics, I believe, will be perhaps it'll be certainly one of the first areas in which you start to see hundreds of thousands of genomes sequenced in secondary care settings. And the other area, which may even exceed the pediatric diagnostic area, is the sequencing of tumors and normal DNA in cancer patients. So this figure on the left, very complicated figures from a review by Bert Vogelstein. Science had a whole special issue on this topic, and in his kind of overview, he regards about 140 genes to have now been identified, which are recurrently and somewhat commonly mutated in cancers and significantly affect disease progression. So the significantly affect disease progression often involves elements of judgment. That is, in some cases, we have a very detailed knowledge of what these mutations do, in other cases less so, and are more reliant just on the correlation between presence of these mutations and behavior of these tumors. That's quite a long list. As we know, cancer is an incredibly heterogeneous disease, and it's heterogeneous in all respects, and certainly in terms of the prevalence of these so-called driver mutations. Some cancers have very few. Pediatric cancers typically have very few. There hasn't been enough time in children for a lot of these mutations to accumulate in their somatic tissues. The adult cancers are extremely variable. I'm going to largely rely for my examples on one of the simplest cases, which has been studied very intensively because it's a relatively simple case. It's acute myeloid leukemia. It has the lowest number of mutations of this type of any adult cancer that's been carefully studied. And as we'll see, it's amply complicated nonetheless. So here we take the list of the genes, a subset of those 140 genes, which are relatively commonly encountered in mutant form in the tumor cells of AML patients. So each line here is a patient. So this study involved a lot of patients, hundreds, and a connection between one of these bars and one of the others means that both of those things were found in that tumor. So the point is that there are some that are more common than others, of course. There's a very complex kind of combinatoric situation in which you see these driver mutations and different combinations in different tumors. And keep in mind, this is the simplest of the intensively studied adult cancers. There's no question that the data coming from these studies affects the behavior of the tumor and in particularly therapeutic response. So here we've done a very crude kind of categorization that has taken a couple of the more common situations on the graph that we just looked at and look at the relapse-free survival of these patients. So it's not great, but it's a lot better than what you see with patients with other genotypes. And there's roughly equal numbers of patients that are in the blue-yellow group on the one hand and the black group on the other. And already, this type of data is having substantial therapeutic implications. One option for leukemia is developed here in Seattle, bone marrow transplantation. It can't be done during the acute phase of the disease. The patients have to be in a stable remission to have any realistic chance of surviving bone marrow transplantation. But patients with these genotypes, which have poor relapse-free survival under conventional therapy, are increasingly choosing to undergo bone marrow transplantation if they can meet the criteria for this demanding therapy. And patients with these genotypes are more likely to hope that they end up in the 60% or so that have stable remissions or reasonably stable remissions. Charts of this type could be shown for many types of cancer. The literature just in the last year or two has a flood of data of these type, and oncologists are struggling to understand how to deal with its implications, but it is infusing kind of new energy and optimism into cancer research, which is much needed. So now move on to therapeutics. The therapeutics I want to really emphasize. The big problem in AML is we don't have very many therapeutic options. It's true for actually most diseases, most serious diseases. We don't it's a nice idea that genomics is going to allow very fine subdivision of diseases in this elaborate taxonomy, but it doesn't do a lot of good to, let's say, take type 2 diabetes and subdivide it into dozens and dozens of different conditions when there's basically only one treatment. And that is a challenge. We need to focus on therapeutics. And some of my comments here will be a little unorthodox, but I believe we need unorthodox thinking about how to adjust priorities within the biomedical research enterprise to increase the focus on therapy and allow, I don't want to overstate this, but allow our strong suit of diagnostics to evolve as it will. I'm not concerned that there isn't going to be a lot of genomic diagnostics done over the next decades. I am concerned that therapeutic opportunities are not going to receive enough research attention. They're more difficult, they're riskier, they don't fit as comfortably into the incentive systems and so forth. So I'm going to talk a little about therapeutics in cancer. So Gleevec is a well-known pre-genomic prototype for a anti-cancer drug that targets an enzyme whose activity has been elevated by a driver mutation. So this is an instance of having too much of a particular kind of protein present in the tumors, and small molecule inhibitors of this were developed, and Gleevec is the trade name for a spectacularly successful drug. Around 2000, Novartis, who developed Gleevec, started running many ads of this type, and I actually knew two people that sort of fit this profile. It's not so uncommon. These people were dying. They were within months of death, and went into amazing remissions. I don't know, of course, whether this patient is still alive, but many of them are, even from this first generation of Gleevec responses. And now, with second generation drugs of this type and more experience in deploying them clinically, the survivals are excellent, and the remissions are long-term, to the point that if you read the literature of this form of leukemia, CML is called, you'll see that a major controversy today is how long should you treat patients. These are quite powerful drugs. They don't have the side effects of traditional chemotherapy, otherwise they couldn't be tolerated long-term, but they do have side effects, and they're starting to look at patients 10 years out that are healthy, have no pathological trace of cancer cells, and it's unknown how long they should continue to treat. This example is incessantly cited in policy circles and to illustrate a whole variety of lessons. One indisputable one is that this is a long, slow process. The initial scientific insights were in the 1960s, and the first human results reported 40 years later by this drug, and now in 2010, this is the standard of care for CML, and the prognosis for CML patients is spectacularly improved.
How expensive is this treatment?
That's a good question. It's expensive. Does anyone know the actual answer to that question? Roughly what it costs per year to keep a CML patient on Gleevec? Certainly tens of thousands of dollars, but I'm not sure whether it's low tens of thousands or mid tens of thousands. It's expensive. And these costs, of course, they're the way we allocate the costs in our healthcare system. It's not expensive to synthesize this drug, to put it in bottles and so forth. It's the amortization of the R&D costs that are the issue. There are about 5,000 CML patients a year in the United States, so it's not the amortization is over a modest number of patients. I don't think Novartis has made any serious money on this drug. They may have lost money on it because the market is small, but it put them in a leading position in a very active area.
It's $92,000 a year.
92, so it's high tens of thousands. So this has stimulated a tremendous amount of work because many of these driver mutations are in the same general class of proteins that the elevated protein in CML is, and similar kinds of small molecule drug development can be done. And there is actually quite a lot of success in the sense that you do genome sequencing of the tumors, you identify driver mutations, you use experimental drugs, patients respond to them, but it takes a long time and very fortunate circumstances to get from that situation to a Gleevec-like situation in which one is really getting excellent outcomes for what was once a fairly uniformly fatal disease. So I just give one example. The literature is also full of cases of this kind. So this is a patient who had this has started the experimental therapy, had failed conventional therapies for a widely disseminated melanoma, was within weeks of death, and had a genome sequence, a particular driver mutation, which was first described in 2002, was present and known to be a major player in melanoma progression. With an experimental drug, much of a Gleevec style, there was a spectacular remission in a few weeks of treatment, but it wasn't stable, and that tends to be the picture today. The interesting point, and the reason I'm showing these somewhat upsetting pictures, is to illustrate that the pattern of disseminated lesions in this patient after relapse is essentially identical to the pattern before the treatment started. And what that implies is that there were pre-existing resistance mutations in essentially all of these metastatic lesions. And that is going to be a major challenge. Resistance was a major challenge in the early years of Gleevec. And there were patients that went down this same depressing path, but with second-generation drugs, which the protein, the gene that goes through this protein cannot so readily mutate around, and with different combinations of drugs that attack the same protein, now we have stable remissions. This is what high school students. This is what clinical research looks like during the stage when you're first getting a foothold, and there's exhilaration, but there is also plenty of heartache. So I want to come back to what some of you will know to be a kind of hobby horse of mine. I've been talking about for at least 15 years at the medical school, which is the notion that genomics might be able to empower less traditional approaches to drug development that could lead to major advances in therapeutics. With respect to the example I just gave, the BRAF driver mutations in melanoma, there what we need is what I would call conventional drug development. There's a lot of it going on. I will just say in passing that there are serious policy issues there. One of the major problems is that every major pharmaceutical company and many biotech companies are churning out these experimental drugs. It's obvious from the patient that I just showed, who's not at all untypical of the good results in these early clinical trials, that we're going to need to treat them with cocktails. I mean, this is the way that AIDS is now uniformly treated. There you have a virus which mutates at a ferocious frequency, but can control long-term these infections in most patients with the right combination of drugs, basically by making it less and less probable that the combination of mutations will occur in one virus or in one cell that will give resistance across the cocktail. That's what's needed in cancer, is a major kind of combinatoric effort to try to couple the genomics to this expanding repertoire of small molecule therapeutics directed against driver mutations. And there are major obstacles to doing that. Sounds like a sensible idea, but the intellectual property issues, for example, are huge. These cocktails you would like to constitute involve drugs that are in the hands of various different pharmaceutical companies, and it's sort of unclear in whose interest other than the patient's it is to actually put all this together. And patient interest doesn't always come first in our capitalist system. But here we are. I'm now going to shift gears to the notion that there are other approaches than conventional drug development, and there are now, I think, increasing indications that their time has come. So my favorite candidate is exploitation of what I call conditionally beneficial loss of function mutations, CBLOFMs. So the notion here is that, so we've seen one path toward developing new drugs is to find mutations that cause some disease or disease condition, try to study the mechanism by which that happens, and hope that some insight will emerge as to how to intervene in that. But you're finding this basic point that I made very early, is that the mutations typically are interfering with protein function. Gleevec is a success story because in that instance the protein is overactive because of the mutation. But the more common situation is that the proteins are underactive or absent. And so drug is not going to bring back this missing function. You need some other lever. And the notion of these conditionally beneficial loss of function mutations is that there are mutations which actually improve people's health, or at least conditionally improve people's health. They're actually rather common. This is an old idea, and just to give credit to some of my predecessors, this is a quote from Science 15 years ago, which captures exactly the game plan here. Identifying a loss of function gene defect. So this is a mutation that causes loss of protein function. That is the physiological opposite of the disease one is attempting to treat. That is, you're not studying the disease, you're studying its physiological opposite. I'll give some examples. Allows one to identify a target that is theoretically amenable to pharmacological intervention. That is, sort of make a drug that will mimic the mutation and get these conditional benefits. There are actually lots of conditional beneficial mutations in the literature. These are just two recent examples from the medical literature. High bone density. This is Rick Lifton's lab that analyzed this case. So this just illustrates some of the issues in identifying and analyzing conditionally beneficial mutations. Is that the proband, as they're called, the person who first called this syndrome to medical and scientific attention, had been in an automobile accident and suffered significant trauma. Was taken into an ER and x-rayed. And so the first surprise is that there were no fractures because there was a lot of soft tissue damage, and the radiologists expected fractures. The second surprise was that the x-ray machine didn't seem to be working because the radiologist had never seen so much contrast between the bones and the soft tissue. There's something wrong with the instrument. It turns out that it just has very dense bones, and indeed they run in the person's family. And but they're not brittle. The auto accident illustrated that. In fact, there was an interesting follow-up study in which someone went and interviewed all these people in this extended family and asked, were they aware that they had these dense bones? Did it have some bad effect that they knew of? And the only consistent complaint is that they had trouble learning how to swim because they could stay afloat. So this is a German child that, as far as I know, remains unique in the world case, who is total loss of function in a gene called myostatin, which is involved in the regulation of muscle development and is just extremely muscular. This child actually had some mild muscle contraction, spontaneous muscle contractions in the neonatal period, but they resolved spontaneously. And now the only thing that's sort of obvious about this kid is he can lift up these barbells and punch it out with his pediatrician. But so the point is that low bone density and and various muscle wasting diseases and whatnot are all physiological opposites of these conditions, and but these are loss of function mutations, and they provide in a very direct way potential targets. And I think this idea is finally going to take serious hold. A very much publicized example of this the last few years was the discovery of a genetic variant in the PCSK9 gene, a protease that interacts with the LDL receptor. The loss of function, heterozygous loss of function, is fairly common in American blacks, very rarely seen in people of European or Asian ancestry. And as it happened in this study, Helen Hobbs and her collaborators, there was actually long-term outcome data on these patients. So retrospectively, you could ask not just what their cholesterol levels were, for example, but what the frequency was of outright cardiovascular disease. And is dramatic protection, having even heterozygous mutation in this gene gives dramatic protection against heart disease. And they've now tracked down neither of these individuals is in the scientific literature, as near as I can tell, but they're much discussed. Two homozygous individuals, and they have virtually no LDL, and they're healthy. So this is a cardiologist's dream, and a biotech company's kind of inspiration. This is an article just from this month in the New York Times. Gina Colada on the job again. So this is a factory that Amgen has built. It's the largest monoclonal antibody production facility in the world, presumably to manufacture a drug for which they've not even yet applied for FDA approval. So theoretically, when clinical trials are underway, there's a high level of secrecy and blindedness and double-blindedness and so forth. But when the results of clinical trials are sufficiently spectacular, as they were with Gleevec, for example, and obviously are here, it's impossible to suppress discussion. And this monoclonal antibody, which lowers the concentration of this protein, the functional concentration of this protein, evidently can kind of drive cholesterol in many patients down to arbitrarily low levels. And the main discussion now is how low it's actually safe to go. If you have someone with very high cholesterol and had a heart attack at age 40 and so forth, there's a temptation to treat very aggressively, and it'll take time to figure out how aggressive is appropriate. But Amgen's confidence is clearly high that this is going to work. So, an abstraction of all of this. This is a modified version of a slide I showed at a psychiatry grand rounds at least 10 years ago. The idea hasn't changed, is that, if you consider phenotypes generally that correlate with health in some fashion, so healthier is out here and less healthy is over there, we imagine that there are going to be some distribution of genotypes. And this is a logarithmic scale, so these are the very common ones, and these are rare. So this is sort of the traditional domain of the medical geneticist, Mendelian diseases and whatnot, and these are very intensively and productively studied. I am certainly not arguing that we should not continue the very effective study. We have a Mendelian disease center here at UW, and we're learning a tremendous amount by studying these conditions. These near normal genotypes, I think is clear, have been overstudied in recent years. There's been a fashion that's a little difficult for some of us to understand for attempting to learn something that's diagnostically or therapeutically useful from looking at near normal genotypes. But a really neglected frontier are these genotypes like the ones that I've given examples of. A major problem in moving the whole system in this direction is just that we don't really have the infrastructure to do that. The academic medical centers had a good thing going for several centuries in which they could sort of set up shop, increase their research intensiveness and so forth, and wait for very interesting subjects to appear for treatment. And that's really kind of what we do. That is, people, they get sick and they present for treatment, and some of the cases are interesting and informative. People that are just having a little trouble learning how to swim because their bones are so dense, just are not showing up on our orthopedic services. And we need, other ways of acquiring these patients. But the difficulty of acquiring them is not a reason not to move forward aggressively in this area. If we had put a small fraction of the resources into this initiative that we've put into studying near normal genotypes, we'd be seeing very impressive progress by now. So I'm going to close with just a few comments about policy. The precision medicine report's already been mentioned. There was a lot of discussion about the title of this report, and it was Steve Galley, the chairman of pathology at Stanford, who suggested precision. None of us like personalized, which is the sort of common term. And the point here, in policy areas, everything matters. Death panels, you need the right name for whatever it is position you're taking. We wanted to emphasize that the thrust of genomic medicine cannot be to go back to anecdotal medicine. Here's an interesting case, different than all my other cases. I treated it different than all the other cases, and so forth. And some of the discussions of personalized medicine are alarmingly of that character. Indeed, there are prominent examples, which I won't go into, of much publicized instances of just this sort of anecdotal interpretation. As David Goldstein at Duke says, every genome has a lot of narrative potential. And we are not interested in the narrative potential of the genome. We want to do taxonomy. We want to get more coherent subgroups of patients and to be able to target more precisely the therapies. But we need a lot more therapies, and it's not a minor matter to redirect an enterprise as large as ours in that direction. So some of the recommendations of this report were sort of self-evident. I've emphasized genome sequencing, but there are lots of other sources of information that should also be collected. But the key thing is we're going to need to study enormous numbers of patients compared to past histories, Framingham-type studies, and so forth. And the only option is going to be to shift towards studying patients as they're embedded in the ordinary course of healthcare. It is simply not an option to do Framingham-type studies on 10 million patients. It is not going to happen, and we need to figure out how to use electronic medical records in the ordinary course of healthcare as the kind of source of long-term follow-up on these patients. And we need much more open source, open access kind of information commons. So this report is full of complicated sort of fancy diagrams. They're actually not very complicated about this information commons, knowledge network, and so forth. The only thing I really want to emphasize is that the organizing principle of this sort of Google Maps-like approach of attaching everything we know to something. In Google Map, it's a latitude and longitude. Here it's going to be the individual patient. That's really the key. We do not presently organize what we know about biomedical information around the individual patient. We organize it in all sorts of other ways. Going forward, the patient is going to rule. We're going to need millions of patients, and we're going to be able to organize a lot of information around them. The co-chair of the Precision Medicine Report, Sir Desmond Hellman, Chancellor of UCSF, was a major player while at Genentech in the development of Herceptin, which has been a very, very effective sort of genomic medicine for treating various types of cancer, especially breast cancer. I think stated this very well in an editorial in Science Translational Medicine, that what we need is kind of a new social contract in which patients both contribute personal clinical data and benefit from the knowledge gained through the collaboration. We've sort of created a system in which the model is that the patient shows up and gets stuff from the academic medical center. And if we're lucky, we might be able to enroll someone in a particularly interesting case in some clinical trial and so forth. But it's not thought of as a social contract in which the quality of what the patient gets out of the healthcare system depends on what their willingness to put information into it. I believe the public can be educated along these lines, and that we need to start that process. there are, of course, immense challenges associated with privacy and many other issues, incidental findings and so forth. But we start. My two injunctions as we leave is that we don't need the hype. We never needed the hype in genomic medicine, and we most assuredly don't need it now. We have real stuff to talk about. This is just a sort of a dot-com era example. The hype got fairly extreme. At the top of this ladder is a world without disease and so forth. We don't need this. It doesn't help us. We have a solid product to sell. And the other injunction is we need to build the commons. And my view is that business as usual is actually not going to get us there. There are many obstacles, but it's good we have some young people here. And we need to sort of gradually not take no for an answer. This is, I think, the path, and I think the benefits will be substantial. Thank you. So do we have any high school students that are brave enough to ask a question here? No? I don't want to put them on this.
We can take a few questions now, and we also serve refreshments afterwards, and Maynard's willing to stick around for a little bit to answer additional questions if you have them. But if people have a few questions now, go ahead and ask.
Not a high school student, but we'll start here.
For a layperson, could you talk a little bit about biochemical pathways? I've seen these big charts.
Yeah.
And the second question is, the Institute of Systems Biology, how close are they to realizing their potential with the 4 Ps?
Yeah, so I can't comment about the institute itself, but it's pretty obvious from the way that I framed my discussion tonight that I'm a skeptic that systems biology is ready to have much impact on the way that we deal with these issues, and it's because I think the problems are, there are a lot of interesting things happening in systems biology, but if you even take the mutations that we sort of think we understand the best, say cystic fibrosis, we have absolutely no ability whatsoever to predict, because this particular ion channel is mutated in these kids, that they would end up with a disease with these very complex kinds of characteristics that CF has. And the same thing applies pretty much across the board. Take BRCA1. Mary Claire may want to speak up here, but I'll take the risk of saying a fairly standard thing, which is, so we actually, after 15 years of very hard work, we actually have a reasonable idea, why mutations in BRCA1 might increase cancer risk, but a very poor understanding of the tissue specificity, why this is breast cancer and not various other kinds of cancer. And those are the simplest kinds of questions. To try to put it all into some very complex kind of engineering type system, I just think is not going to happen anytime soon. Doesn't mean there aren't interesting things to do with systems methods, but I don't think this is one of them. The metabolic pathways is, we're getting better at them. The charts were already complicated when I was a student. It's one reason I went into inorganic chemistry instead of biochemistry. These charts were too complicated for me. They've gotten a lot more complicated, mostly because we have not actually discovered a whole lot of new pathways since I was a graduate student. But we've come to understand the complexity of many of the pathways to a sobering degree. For example, there are often, we used to think there was one enzyme that got you from here to here. We now know there are 40, and that some are expressed in the pancreas, during the first trimester of pregnancy and so forth. It's a very complicated situation. This is critical knowledge. My Beery twin example, the inference from the genome sequence to the therapeutic depended on knowing in considerable detail how dopamine and serotonin are made, and many applications are of that nature. Genomicists love biochemistry. It's just a field that progresses more slowly than we would like to see it. We don't, just don't learn that much new, but we get as much mileage as we can out of what we've got. So, back there, yeah. So if I understand the question, it has to do with, is moving down this path likely to increase health inequities? And is it that the type of concern? Yeah. That we already have that sounds expensive and involves a lot of really specialty medicine and so forth. And the Berries were upper middle class, highly educated family that knew how to go out and get the best they could for their kids and had the resources to do it. It's a concern. I suspect my reading of the history of medicine is that relatively well-off, well-connected, well-educated people have always been the first adopters and the first beneficiaries of major medical advances, and that'll probably happen here too, which means that we need to use explicit mechanisms to try to compensate for that natural effect. The PCSK9 story is quite interesting because this the African American population is certainly understudied genetically, particularly because it's genetically more complex than individuals of either European or Asian ancestry, and this is a West African founder effect mutation that underlies this phenomenon, and there happened to have been a study in Dallas, I believe, that had done this long-term follow-up and had major African American participation. Otherwise, this wouldn't be happening. So we would like to have it is indeed critical from a basic science point of view, as well as a social justice point of view, that there be intensive study of all of the diversity of the population. The U.S. should try to turn this to its advantage. And we're a rich enough country that we could, if we had the will. It's great to study the Icelandic population. I'm going to go to the Faroe Islands in September, where they have an even more narrow kind of population, and there are major benefits to doing that, but they are outweighed by the benefits of studying very diverse populations. The latter activity is just much more expensive and more challenging, but that's our society, and that's the way we should go. Back there. Yeah.
What do you think is the role that DIY bio and citizen science will play in the building of the data commons for genetic and genomic data?
I didn't quite get the who will play?
That citizen science.
Citizen science,
DIY bio.
So this is, posting your health records on the web and finding a similar group of patients and no.
Doing biological experiments.
Wet lab experiments, yeah. Well, I would defer to you about that subject. I think that most of the people in this room, most of the graduate students here don't even do wet lab experiments anymore. They just sit at a computer terminal and analyze data. And so I think wet lab experimentation is going to become actually a more rarefied activity, even in academic medicine. And so I'm skeptical that there's going to be an outgrowth of citizen science in this area. What I'm sure we'll see happen, and we're already seeing happen, is that at the more informational level, these so-called disease interest groups, groups of spontaneously assembled groups of people that care deeply about some potentially rare disease, they share information. And that will include genome sequences and clinical information and lots of information. And that we'll start to see that to be a substantial force and could indeed become a creative one. I mean, people are cynical about like the Lorenzo's oil kind of stories because they often don't hold up to close scrutiny. But a lot of stuff that we do doesn't either. And as the groups get bigger that are sharing data and more disciplined and so forth, we may see significant citizen activity that. But I doubt that they're going to be doing a lot of wet lab experiments. So just my guess. So we're going to have a reception.